very much. Buenas tardes, señoras y señores. Sé todo. Sí, claro. Muy bien. It's a pleasure for me to be here, so I like our hosts who invited me. And as was mentioned, I've been the director of the Department of Medical Oncology and Hematology in Freiburg until a few years ago, for almost 25 years. But I've been involved in seeing cancer patients for almost 50 years now. And that certainly gives you a little different perspective on many things. So the title of my talk, of course, is a little unusual. So I base all my experiments, be they in the wet lab, in the computer, or in my brain, on clinical observations, evolutionary interpretation, and trying to use digital intelligence, machine learning, to take it one step further. But I would like to make one statement that medicine and science, in my opinion, are inseparable. For me, medicine is a natural science, with more implications, of course, like all fields in science. So I would strongly argue always that we have the biomedical exchange program. I would never, from my perspective, exclusively focus on one part of the field and not the other. The second statement I would like to make, that in my opinion, research and teaching belong together. As some of you know, that's a classical German Humboldt tradition since the early 19th century. So when we talk about science, I think we also always have the students in mind. So far for the introduction. And just as one example, how putting science and medicine together, we actually heard this morning about the young pediatric doctor, I can't remember her name, who talked about autophagy, apoptosis. She's a clinical pediatrician doing cutting-edge research. And this is just one example from my own history. When I was in New York, our topic was studying normal and malignant hematopoiesis. And we discovered a factor that regulates the production of neutrophils, GCSF, which then has become a drug, Neupogen, which is extremely effective in treating and preventing drug-induced and congenital neutropenia. So this is to support my argument that medicine and science are inseparable. So the approach to the problem where we do stand with cancer today is to review the clinical and experimental evidence, to put that together into perhaps somewhat novel concept. The next step is to transfer this concept into a mathematical model and do a few experiments with it. And this is where we are at the moment, trying to take the mathematical model into a laboratory, back into the laboratory to do experiments. So what is so special about cancer? You always get many answers when I ask the audience, what's so special about cancer? It can kill. Other diseases can also kill. Yeah, there are other diseases we don't know how to cure. It's not one disease. That's a good point. I think why it's really frightening, yeah?
Because it has its own life. And it's
Exactly.
life within us.
And it starts from our own body. A cell suddenly decides to go a different path. And that's why I think this title is quite well chosen. The Emperor of All Maladies. This is a surgeon who worked at the NIH in Bethesda. It's a brilliantly written book, but it sort of makes it clear why there's something different about cancer compared to other diseases. And the question that we always hear, of course, is why is there cancer? Why do we get cancer? And the person who first understood why we get cancer is Heraclit. Anybody knows who that was? Pedro, you should know. Right. Famous Greek philosopher. There were actually two schools. His school was that everything flows. Nothing is stable. And that, in fact, is true. And this is the famous Brownian motion. Let's see, how do I... And I really like this video because you see these cells moving, getting together, then separating again. This is what happens in our body this very moment. And it's not just molecules, it's also cells. There are similar videos of cells, how they go around, move here and there. Incredible activity in our body. And this, of course, generates a conceptual problem, which means every experiment that we do is just a snapshot. So if you do the experiment now, and you do it 10 seconds later, the result will be different. And that is probably part of the reproducibility crisis. That's big discussion that even people in the lab cannot repeat their own experiments. And this is probably a part of the problem. And what drives this, is, of course, is energy. And the next step, so everything moves, which also means everything changes all the time. And this is a nice collection of numbers that I got from Christoph. We have about 10 to the 14 cells, 200 different cell types, and 1 million cells divide every second. Bang, 1 million, bang, 2 million, bang, 3 million. So there's incredible things going on in your body. But when cells divide, obviously the DNA has to be duplicated, and that works extremely well, but it's not perfect. So we have to expect about 3 million genetic mistakes per second in our various organ systems that have dividing cells. Most of the time, this doesn't matter because the mutations occur in irrelevant base pairs. But of course, what is very clear, the older we get, the more mutations we accumulate. So Pedro, you and me are a little older, so we have a lot more mutations than those young guys. So that is unavoidable. And the driver, of course, is entropy. And Vogelstein put that into a nice intellectual experiment, these observations. He looked at different organs in the human body, and there were data available how many cells divide every second in each organ. There are big differences. For example, heart muscle cells, how often do they divide? Never. So do you get cancer there? No. So what they found is in an organ with a low division rate and a low predisposition, genetic predisposition to mutations, you rarely get cancer. In an organ system with a lot of cell divisions, especially when combined with a high mutation rate, you get a lot of cancer. And they came to the conclusion that 30% of all cancers occur as a result of random mutations. Just because of the cells divide so often, there's a certain incidence of mutations, 30% of cancers. And that actually resulted in a lot of uproar in German newspapers. There's nothing we can do about cancer. It's just fate. It's terrible. So he picked up on that point. And he just, I think 2 weeks ago, he changed his model somewhat. that we have here the normal cell, stem cell, because the cell has to be capable of dividing, otherwise you will not get cancer. And then the different stages until you finally have a clinically relevant cancer. And here he says at each stage you have random mutations, errors, which account for about 30% of all cancers. And then you have environmental factors. And what we have learned this morning is that environmental factors lead to epigenetic changes in the cells. But cancer is a genetic disease. So how do epigenetic changes translate into mutations? And there was a very interesting paper just at the end of last year where they have shown that epigenetic plasticity and genomic mutations are related. And as you might probably know, Lamarck always said that acquired properties will be transmitted. Darwin said, bullshit, only genetic changes will be transmitted. But what they found is that in many different organisms, like lizards, for example, if the epigenetic response is on for a long time, it will eventually translate into a mutation in that gene. So epigenetics and genetics are not completely separated. The practical example is you have a lizard who can regulate the color of its skin. So it makes it very dark when the surroundings are dark. But when the lizard stays in that dark area for 5, 6 generations, then he will stay black. And you're having mutation in that gene, turning it on permanently. So our thinking of epigenetics here and genetics there is probably no longer valid. So we have stem cells, mutations, evolution of cells, epigenetic factors. The question is this clinically relevant? And in this very nice paper in Nature, it's actually sort of a brief review, it's a very interesting translation of this knowledge into clinical medicine. So here you have the first mutation, then these cells acquire additional mutations. And it will, of course, be very difficult to have specific, individualized treatment that catches all these different mutations. So I'm not as optimistic as some other people are that we will be able to cure cancer by a totally individualized approach. So we have to think functionally. So what happens? Here in this example, we have the green cells are cells that are sensitive to the chemotherapy that I use. The red cells are resistant. When I treat the patient, suddenly I have very few sensitive cells and a lot of resistant cells there. And if I continue treatment, all that's left over are resistant cells. So in some patients where you cannot cure the disease anyway, it might be better to stop here and give the not so ugly cells a chance to regrow and establish a balance. So it's adapting for balance, they call this approach here. This is clinical oncology, where these findings are relevant to. Another aspect is, do you have a patient with sensitive cells again and one resistant cell? You treat the sensitive cells, and the resistant cells are left over. In some cases, it's better to just stop treatment here, and after 6 to 12 months, the not so ugly cells will reappear. So I think our clinical approach to treating cancer is profoundly, or should be profoundly influenced by these new data in basic oncological research. So I talked about mutations, stuff like that. So it's all an intellectual experiment. So the next question is it for real? And there was a very interesting paper published in 2014 where they looked at the blood from different people in different age groups. And we're looking, are there any clonal blood cells that have mutations? And in fact, at age 20, like most of you over here, no mutations. But when you come closer to Pedro and me, about 15% of the patients carry mutations. And at age 100, 30%. And this increase is exponential. And that is probably very important. It means if you have already one mutation, it's more likely you get another one. Cancer evolution is exponential. So this is clearly age-related. It's logarithmic increase with age of these clones with mutations. And those patients that have these mutated clones have a higher risk of developing leukemia. Make sense? Absolutely. But then the big surprise, these patients also get more heart attacks. And it's not completely clear why. Andreas and I frequently discuss this aspect. The most valid hypothesis is probably that it has to do with an increase in inflammation, whatever the mechanism may be. And that is also important for medical doctors. So here you say, oh, that's a blood disease. No, it's not a blood disease. It's a disease that infects the whole body. And that's probably also an important message for the future. Here another example from the clinic. Patient with leukemia. The gray one is the dominant clone. Then after a while, new clones appear. Patient gets treated. Treatment was not very effective. So the old clones reappear. And also a new clone appears, which is much more aggressive than the previous clones. So that is relevant for, I think, a lot of clinical decisions. But it's not only the blood. This paper I found really a little frightening. This was in Science at the end of last year, I think about October, November. Martin Corina and her colleagues from Cambridge in the UK, they studied the epithelium of the esophagus, collected cells and investigated them. Is that clear? Took a little biopsy, looked at the cells. At patients that were young, actually these are healthy people, 20 years, 75 years. Each dot represents a mutated clone. So even you young guys at age 20, you will have a few mutations in your esophagus. And I hate to look at this picture. It's not only the number, the size of the circle also indicates the size of the clone. So when you look at these pictures, you think all of us are going to get esophageal cancer. But that's not true. Here we carry a lot. These cells carry driver mutations of which we previously thought driver mutations make cancer. These have a lot of driver mutations and don't get cancer. So our thinking about driver mutations causing cancer is far too simple. So this is what I've said before, just explained. We have the esophagus. We collect multiple driver mutations during your lifetime. And to get cancer, something else has to happen. And we have no idea what that is. And in this review, the conclusion was these findings suggest that transition from healthy tissue to cancer may be more complicated than we have modeled. So we have to rethink a lot of our cancer models. And this is also illustrated here. We heard this morning, we have 20,000 genes, which can have 30,000 post-transcriptional modifications just on the messenger RNA level, probably another 10,000 modifications on the protein level. So basically, we are talking about billions and billions of components. And it's very unlikely, by just studying one or two of these, we'll understand really what cancer is. So this is time to sit down, have a glass of Malbec, and think. And see how we can put these observations together into a model. And I propose that we should not that we should stop looking at molecular factors, but I think studying the molecular pathways have reached a certain limit of what we can learn from them. So we should probably look at functional pathways. And we have seen this picture before this morning. This is what Hanahan and Weinberg did. They did a huge review of all molecular cancer literature and grouped the results into different pathways, like evading growth suppressors, enabling replicative immortality, and then suggest we should try to target these pathways as therapy. So we thought that just perhaps takes us a little further. So we decided to develop a computer model, a simulation, where actually we started about half an hour ago with one cancer cell. Yellow ones are the ones that are dividing. Red are the ones which are destroyed by the immune system. Blue are cells that die by apoptosis. They haven't reached that point yet. And you can play with this. You can increase immune destruction. You can increase growth factor signaling. So this is like a tool, like a game that you can use to better understand cancer. So we took the ideas from Hanahan and Weinberg, put them into algorithms, and where we wanted to have a real-time observation and interactive, so you can, let's say, avoid the immune destruction. You can give it less replicative immortality. You can increase growth factors. So we learned a lot by just putting our clinical observations into a mathematical concept. And then we decided, okay, we get a computer, a fairly big computer, and let the computer run 1,000, I think 1,100 experiments in that model by just pressing on these buttons in a random way, because there are billions of possibilities. And then we investigated the results of these 1,000 experiments statistically, and it turned out that there are only 3 pathways that are really important for cancer cells to develop, at least in this model. And this is the acquisition of immortality. Normal cells die after 50 to 70 divisions. Oopla. A preferential growth signal affinity, and it doesn't really matter whether you decrease inhibitors or increase stimulators, the net effect counts. And you always need a block of differentiation and stem cell properties to allow the cells to divide. And we were quite happy to see another paper by Bert Vogelstein, where he defined the cancer pathways by molecular research. So we just did an intellectual experiment. And they basically came up with almost identical conclusions what the relevant factors are about cancer. So now the next step is, of course, to try to take this back into the lab to see whether our model is valid. But here we encounter the problem that the colleague who did the statistical analysis this morning also addressed. Here I'm just talking about 10 parameters, 10 concentrations, 10 time points, 10 to the 100 experiments to do. It just cannot be done. And after I heard Dr. Kornblitz's talk, I mean, if you just look at the messenger RNA variants for one gene, plus the protein modifications, this just simply cannot be done. There could be a unique combination of all these factors, which is perfect for cancer or for killing cancer cells, but we don't know it. So I think that modeling and simulations and using machine learning might help us to find the one important needle in the haystack. I don't see any other chance that we have. So I worked in these computer models with a group of brilliant young guys who work in machine learning. And we talked about this morning at artificial intelligence, but I just would like you to illustrate what it means. So the task here is to get the pendulum upright. So this little sleigh just moves up and down, tries this, tries that, tries that. I think you probably couldn't do it by hand. So after 236 trials, the guy knows how to do it. So, which means here are also thousands and thousands of variables that play a role in this process. But using evolutionary algorithms, you can detect and identify the most important parameters. So this is our concept that we want to use in cancer research. And just the second model, which really was the clue for me, the same group was world champion in robo-soccer. So these are little robots that play soccer. Why did they win the world championship? Because they used machine learning to train their robots. All other groups programmed them, looking at temperature, at friction, at I don't know what. It's impossible. There are too many possibilities. So they said, well, we'll let them learn. Oh, should be. Oh, yeah, it's coming. And they just have a camera, no other input of data. And they told the robot, if you get the ball into the goal, you get a cookie. If it doesn't get into the goal, you get a whip. It's translated, but it basically works like that. So first, this thing just runs around, already has learned to sort of keep the ball next to it. They were not programmed, they just learned it. Now the final of the World Soccer Championship, little guys. Freiburg is advancing rapidly. Look, almost made it. So it's really stunning. It's only a small chip that the little guy, with the only data input being a camera, no other data input, learns how to play soccer, at least in this system. So we thought, okay, if a camera is enough to get all the data you need to learn something new, we thought, why don't we use that for experiments related to cancer? So the first project we have started is to optimize drug dosing and schedule for breast cancer cells. The clinical goal, of course, is to minimize cell number. And the second project is to improve T cell growth and gene transfer into gene cells for CAR T cell production. Here we want to maximize the cell number. And you need, of course, vessels that are good for this type of an experiment. So these are the classical culture vessel. We want one with an inlet and an outlet, so we can constantly change the conditions. This chip is almost 6 centimeters long and 2.5 centimeters wide. So it's just like a regular microscope chip. And in this case, has 44 little chambers. And this is transparent material, so we can look at them through the microscope. They have a number of advantages, because what we need is real-time imaging. We want just to look at them with a camera. We don't want to collect any other data of these cells. So we have the chip. We put the cells in there, let them grow. We have 2 pumps. One is medium, and one is drug. And these pumps can be adjusted to run at different speeds. So we want to try out which type of dose, concentration, and schedule is the most efficient one. And what I've heard this morning from our pediatric colleague is that the dose apparently is crucial. If you are too low or too high, you might just get the opposite result of what you're trying to do. And to test this all out in the lab is close to impossible. So we hope that this system will allow us to address that point. Then we have a microscope that analyzes the picture. It's only bright field. We don't stain those cells. Do an image analysis, parameter processing, look at them. That data is fed into the control laptop, which then decides how these pumps should be run. So this is the overall project. In the cancer project, we want to kill the cells. In the T cell project, we want to grow the cells. And this is the experimental setup. It's not as simple as it sounds. This is a fairly sophisticated microscope. The little chip is in there. Here's one pump out here, which sort of feeds the little chip in there. This is the image analysis software, and this is a computer running the pump. So just to give you a practical idea of what it looks like. So we get pictures like this and pictures like this by taking pictures every 30 minutes of all the chambers in here. Pictures every 30 minutes for 18 hours, which is a lot of pictures, in these little chips. And we, of course, are trying to analyze this at the moment in a fairly conventional approach. This is software that's available. It's called Fiji. This is a monochromatic light field picture, which transfers it mathematically eventually into a binary picture, which we, of course, need to feed the computer. And what we can do so far is to measure the area covered by cells as a surrogate parameter for the total cell number. And amazingly, this works not only for adherent cells, but also for cells in suspension. That's probably a statistical thing. This data is then fed into the computer. But we, of course, hope to get better by using machine learning for image analysis, together with Joschka and his team. And the first data I have seen from your group already were stunning for me. So I think we can get much more information from here. Most of you have not heard the talk by Dr. Frank he looks at molecules and takes these electron microscopic pictures in a very elegant way. What we are trying to do, use a very similar technology to get three-dimensional information about the cells. So it's conceptually the same. And then, of course, we hope that after optimizing the cell data, then the machine learning program, hopefully, we find the best way to kill cancer cells with a certain drug. And then it gets really exciting because we can add X pumps. At least, Josha, that's what you told me. We can use X pumps, use the same approach. So we can look at drug combinations, drug sequencing in different sequences. So my hypothesis is that using machine learning in a setup like this might have a major impact on getting better at treating, killing cancer cells or growing cells that you want. And just to show you that it can all be fun, we started to, because it's all important to visualize things, we developed a computer game. Oopla. There it is. And actually the music is the best part. I don't know how to switch that on. I don't know. Anyway, so we developed a system where evolution occurs. Stopped, huh? Oh, the music is so great. Anyway, so we have sort of a test tube here, a virtual test tube with cells that divide, cells that have different properties, and each color, we presume 10 hypothetical genes, and each gene has a color. So when other genes are expressed, the cells change their color. And you can play with the system, controlling conditions and letting cells grow. It's just fun to watch. The plan was that people would really love to play this, but for the game people, it's not fun enough. For the science people, it's not science enough. But all the experiments we did were really based on the experience in this computer game.
Is it on the App Store?
Yeah, you can buy it for 99 cents or so. It's just fun to watch and to play with it. And we just did, I mean, theoretically, we could do more experiments with this game. We just did a few to see what are the properties of the most aggressive cell. Would it have many, let's say, aggressive proteins or no? No, the cells that had the least properties turned out to be the most aggressive one in the system. And that certainly is what the clinical tells us. The less differentiated the cells are in a cancer, the more aggressive the cancer is. So let me just conclude, getting a little bit back to medicine. So we have seen that mutations are part of our aging. And if we say mutations are part of getting cancer, then aging and cancer are an integral part of our lives. I think we have to accept that. And at this point, you can perhaps reduce the risk of getting cancer. If you live long enough, all of us will get cancer. There's one other way to die. A lady from Holland died at age 130, never been sick in her life. And she said, please study my body when I die to find out why I am dying, never been sick. Why did she die? no more stem cells in any of her organs. Because stem cells can divide, let's say, 70 times. If your stem cells have divided 70 times, that's it. So you either die of cancer or of stem cell exhaustion. But of course I think we rarely cure it, but we can always control it. And knowing these new insights into cancer biology, I think will help us to do that. And I think we really have to look at the patient as a friend. You have to picture how you would behave if you were the patient. The worst thing for a doctor is to be arrogant, be humble, know what you cannot do, try to understand the patient's perspective, show empathy, and I have a very strong belief that we are required to learn from our patients, especially when you understand that each cancer is different. Each patient is his own disease in a way. As a responsible doctor, you learn from each patient, because often big studies do not pick up the really important aspects of many diseases. So while we will not solve the aging problem or the cancer problem, I always look at this quote from an American essayist and poet: to know even one life has breathed easier because you have lived, this is to have succeeded. So if we can solve small problems, I think we have done well. So now I just have to say thank you. Of course, there are a lot of people I have to thank you: my teachers, my mentors, my students, my colleagues, my patients. But this is the current team. For the biomedical experiments, I have 2 students who work on T-cell expansion and breast cancer chemotherapy, together with Marie Follow, who is the lab director. I'm just a guest in the lab. I'm nobody. And then a software group with Joschka Bödecker and his colleagues, and a hardware development group, because it's the software we need, the biology, but also we need good hardware to do those experiments. And that's Gustavo Roseo and Ana Pereira, 2 students from the technical faculty in Buenos Aires. Their boss and his wife. We have a cooperation with an Argentine small company that makes some of the hardware for us. And I should add that this problem would not have started without the IMBS program. He approached me during one of the translational medicine conferences in Buenos Aires. So I hope at least one person talks to me today, asks for collaboration. So I think I want to stop here. That working with our colleagues and friends in Argentina has been extremely stimulating, I think extremely productive, and I will hope I will continue for a few more years. Thank you.